Okay, good morning to everybody. I'm Elisa Massi. I'm a master's student in bionics engineering in University of Pisa and Scuola Superiore Sant'Anna. And today I'm gonna present the work of the research of my colleague Martina Finocchiaro and me. And our work is about the real-time neuromorphic encoding of visual stimuli acquired through an external camera in a Labview environment. The main objective is the implementation of an algorithm for visual feature detection. So we want to simplify the problem and acquire an image from a camera. We want to divide the field of view in different regions of interest from which we can extract a feature such as the color, the shape, and the edge, and convert this feature in a spiking trains. In this way, the information will be compressed and we will have reduce and lighter data packets that can be transmitted with different protocols to other devices for a communication of information that was basically to mimic the ganglion cell dynamics, which, as you know, the ganglion cell is the last cell in the visual chain in the retina, and they can transmit information through spiking rates. Another bioinspiration we want to give to our project is based on the configuration of the acquisition of information in the field of view. So we divide the field of view in the camera in different regions of interest. So, and each region of interest is directly connected to a neuron structure. So as in the retina happened, we have an acquisition of feature independent from each region of interest, which can be decoded by a different neuron. In our case, the algorithm wants to recognize color feature. So for starting, we decided to decode the main color, so the three wavelengths that our photoreceptor in the human eye can recognize in the cones, and the black color representing the role of the rod in our retina. This is shortly how our algorithm works. So from the real world, we acquire the information from the camera, and in Labview environment, we overimpose a grid of variable size on the picture. So from each region of interest in the grid, in our case, the feature, which is the color, the main color of this region, is decoded in a firing rate, in a current. This value of current will feed a neural model, which in our case is the Dikevich neural model. And just by developing these two differential equations, we will obtain a voltage dynamic. So in the decoding phase, finally, these voltage dynamics are analyzed in frequency domain. And so based on the frequency of firing of a neuron, we can understand the color, recognize it, and so having a partial reconstruction of the image based on the color. The neural model we decided to use was the regular spiking Dikevich neural model because it's very powerful. Just using two differential equations, the one shown in the slide, it can reproduce 20 neural dynamics, just changing the four hyperparameter you can see. So AB, the voltage of resting state, and D, with changing the dynamics and the behavior of the neuron. And in this slide, there's an example of a real application of our algorithm. So it's embedded our algorithm in an autonomous robotic system for artifact detection on a target object. So we have a serial manipulator robot which moves on the target object and an embedded camera placed on the robot. The camera acquired in real time a video information that will be transmitted using cable to an embedded FPGA board fixed on the camera. And on the FPGA board, the algorithm, the virus algorithm I explained before, is running, and so producing and converting the feature, in our case the color of the video, in spiking rates. And these spiking rates, in the form of compressed information, are transmitted using wireless protocol to a computer where the user can monitor and control all the system. Right now we have reached some conclusions, so we can in real time encode red, green, blue and black color from the real-time image, and with a resolution, we are now, the spatial resolution, we are now working on it, and is increasing, but now is in the demo also is a 5x5 grid overimposed on the image, and with a temporal resolution of 20 kilohertz, which is biologically plausible for the number of neurons that we are using actually. So I want to stress the fact that the neuromorphic encoding is really powerful because allow us to compress the information, the usual information we want to transmit, since we can in this way transmit layer packages of data, and in this way save in power supply and save in energy during the working of our system. And we are now currently working on embedding this algorithm on an FPGA board, and okay, as I showed before, embedded this system in a robotic system, and this system finally can be used in future neurophysiological study or in closed-loop hybrid bionic system. Now, thanks to my colleague Martina Finocchiaro and all the other people in the lab who collaborate. Thanks you all for your attention, and let's see a short demo. Okay. This is a Labview demo showing that the real-time image acquired is decoded by the neuron, and each the different region of interest decoded a different color based on the neural spiking rates. And this is an example and a focus of the behavior of a single neuron representing a single region of interest, now spiking at low frequency, but changing color, spiking at higher frequency. So having a different behavior, so thank you again for your attention.